So today, as you can see, Wigge could join us, and I just want to mention about his scientific story, how it started. So he did his master at University of Oxford, and at that time he was working on E. coli, and he tried to understand specific site-specific recombination in E. coli. Then in Cambridge, he did his PhD. And after that, he moved to Salk Institute when he did his postdoc at the lab of Professor Dr. Wigge. And after his postdoc, he moved to Germany, went also together with Professor Dr. Wigge. They were working at Max Planck, and at that time, Philip already started to have his own small group in the department of Professor Wigge. After Max Planck, he decided to move to John Innes Center when he started his independent research. And after seven years there, he moved to Sainsbury Lab. And now he is a professor, appointed professor at University of Potsdam, and also a head of the department at the Leibniz Institute for the Vegetable and Ornamental Crop in Grossbeeren in Germany. And today he will introduce us to the temperature sensing in plants. So we are really looking forward to your talk.
Thank you very much, Monica. It's really kind of you to invite me. I'm really appreciate that. I'm very honored. And it's always great to talk about what we're doing. So as Monica said, we're now working in Grossbeeren at the Leibniz Institute for Vegetable and Horticultural Research. And I put a little map here. I just don't know if you can see, but if you're ever in Berlin area, we're basically on the outskirts of Berlin and sort of the airport. If you come into Berlin, you'll come into Schönefeld Airport. So we're quite near the airport. And we're also quite near Potsdam and the MPI in Golm. So it's a very nice community here. A lot of plant biology labs in nearby. And what I'd like to talk to you today is really what's been my sort of major focus over the last a decade or so, which is to try to understand how plants sense temperature. Let me see if I can just get the mouse. Let's see, okay, slide advance. So my favorite example of this is a chestnut tree that grows in Geneva. So this is a tree where people have been measuring when this tree flowers for the last 100 or so years. And so they have a very nice data set. And this is just measuring days into the new year for when this plant flowers. And so what you can see is for the first 100 years or so in this experiment, the plant was flowering basically in March or April, so about 90 days into the new year. And then in the last century, basically what you see is this rapid acceleration. And in 2003, that this tree was actually budding in December. And so this is an interesting acceleration of behavior of this one tree and a big change in its sort of lifestyle. And we obviously don't know the exact basis of why this is occurring, but it does correlate with temperature. So during this time, temperatures have increased by about one degree C, okay? And so this is one tree growing in Geneva. So you might say, well, it's just an isolated case. It might be affected by other things. But if you look more widely, there are many examples now. So this is an example of a phenology map. And so this is simply showing how advanced particular flowers are in flowering in springtime. So this is from 2017. But in March of 2017, in this area of the southeast US, or much of the south of the US, basically most plants were almost like a month early in their flowering time. So they were advanced by about a month. And actually this year, I checked this map for this year, and already plants are already actually about 20 days more advanced than you would expect for normal behavior in terms of flowering and bud burst. And that's also particularly surprising because this year, of course, they had record cold temperatures in the US in this area, but plants have bounced back very quickly. So we are sort of engaged in this remarkable experiment right now where we're changing the temperature on Earth, and we're doing this enormous global experiment, which is affecting basically plants profoundly, both crops and wild plants at every level. And of course, because plants are basically the foundation of virtually all the food chains, There are all sorts of knock-on consequences which we could anticipate. So it's going to be really interesting in the next few decades to see how plants and things higher up the food chain change and evolve. So there have already been a number of pretty interesting studies. This is just one example where people found that in the last 100 years, you can go to areas that have been sampled really well. So Henry David Thoreau was a great writer, but he was also a great naturalist. So he had a really good sampling of this area around Walden's Pond in the US. And so he kept really good botanical records of the plants. And so people went back and they checked again. They found a lot of plants had gone extinct and other plants had done really well. And it turned out that this wasn't random. So plants that have done relatively well in a climate change scenario tend to be those plants that use temperature to entrain seasonal behavior. Whereas plants that use photoperiod to entrain seasonal behavior do much less well under a climate change scenario. And this sort of makes intuitive sense. So when climate is constant, you can use photoperiod as a proxy for temperature. But when temperature moves out of sync with, or at least historical sync with photoperiod, then you could imagine measuring temperature to control your life cycle is much better than using photoperiod. So these are the climate stripes, for example, for Saudi Arabia. And as you can see, like everywhere else in the world, there's, particularly in the last decade or so, there's been this dramatic increase in temperatures. So how fundamentally, how does this affect plants? So you could imagine basically most plants have colonized almost every ecosystem in the world. And so some plants will have adapted to much colder regimes. So if you go up to the Arctic or Antarctica, you have mosses and plants that can grow around 0 degrees. And then in other places like Saudi Arabia or in the tropics, you'll have plants that are completely happy to grow at 50 degrees. And so basically there'll be a distribution within which a plant will be reasonably happy. And when it moves out of that temperature distribution, it'll get stressed. And so the point here, I guess, is that slightly counterintuitively, but what is a heat stress for one plant might actually be a cold stress for another plant. So broadly, we can think about temperature in two major ways when looking at plants, obviously. So you could have temperature as representing a stress. So the plant will be exposed to a temperature stress, either heat or cold. It'll then acclimate. Alternatively, you could say plants can respond to temperature as a developmental signal. So this is also a majorly important aspect of plant growth and development. So plants will use temperature signals to decide how to control their life cycle. So this is obvious in things like trees, in northern climates where trees enter a dormant period during the winter, and they protect their meristems in dormant buds, which are very cold resistant. And then of course, when spring comes, they need to open the buds and flower. And that's all basically very much depending on weather and climate, and that's majorly impacted by temperature. So you have these two pathways. You have really stress and development, and both of these are needed to maintain fitness, okay? So when we're thinking about temperature, what are we thinking about? Well, so what is temperature? Temperature is simply molecular motion. So it's the movement of particles, okay? And that makes it rather difficult to study because when you change the temperature, you change essentially the entire setup of the cell and the whole system. So when we were starting the lab, we started with this very simplistic view. We said, okay, so here are two plants. One is grown at 18 degrees and one is grown at 23 degrees. And these are two Arabidopsis plants. But what you can see is that just growing them at 5 degrees C different causes a huge difference in their developmental behavior, gene expression program. So the plant on 23 degrees is much faster flowering, has thinner leaves, it has longer petioles, and it has a faster plastochron and it's early flowering. The one on the right has thicker leaves and it's much slower flowering and slower growing. And we've been basically trying to understand, how does the plant on the left know it's at 23 degrees and how does the plant on the right know it's at 18 degrees? And so if we can understand how temperature is sensed, then we're going to be able to maybe understand how that information is integrated into the plant's development. And finally, the ultimate goal of the lab is to be able to engineer thermosensory behavior in a controlled way, both as a proof of principle, but also because this might be useful from a sort of biotechnology point of view. So we do argue that if we do actually understand temperature sensing properly, we should be able to take a plant and control its perceived temperature in a very sort of straightforward manner. So if we're looking for thermosensors, we should set some ground rules, and these are the ones we use in the lab. So we say if something's a temperature sensor, basically it should have a temperature-dependent physical activity. So it should be the thing that responds to temperature. It should be involved in controlling an important temperature-dependent behavior, right? It should be relevant. So a number of things in the plant cell respond to temperature, but we often can't find them controlling an important response. So they're not so interesting for us. And then finally, we want to be able to tune it to modulate temperature responsiveness. So if it's really the key temperature sensor, if we modulate it, we should modulate that entire temperature response pathway, which it's controlling. And so just to give you an overview, I'll just touch on a few of these things today, just to sort of give you a taste for what we are interested in the lab and what we've been doing recently. So in terms of Arabidopsis, if you go down to low temperatures around 4 degrees or freezing, there are two major pathways that stick out. One is vernalization, which is a developmental pathway which controls flowering behavior, and that's controlled by a gene called FLC. And I won't go into that today. It's a lot of good people are working on that. The other area which is also interesting is this ability to acclimate to the cold. So the plant has a very sophisticated mechanism to induce protective genes that will protect the plant very successfully against freezing. And this is called the so-called CBF pathway, which is I'll talk about later. If you go up in temperature, so higher temperatures, they invoke a whole set of different responses. So a major response of plants to warmer temperatures is something called thermomorphogenesis. So that's what I alluded to earlier for the plant even grown at 23 degrees. As you increase temperature, plants flower earlier, they have thinner leaves, and they grow faster. And so often around 27 degrees, we call this ambient temperature. And what we mean is it's basically a temperature that has a high temperature, is high enough to cause a developmental change in the plant, but it doesn't cause widespread activation of stress pathways. And so for us, historically, it's been very useful for us to study ambient temperature because you can often then see early events in the temperature sensing pathway before large-scale stress responses are activated, which sometimes can cloud the picture. So during nighttime, warm temperature causes things like hypocotyl elongation and early flowering, and this is modulated by a thermomorphogenesis pathway. During the daytime, there's also a very important high temperature stress response driven by the heat shock factors, the heat shock factor, transcription factors, which activate heat shock protective proteins. Okay. And so today I'll just talk about a few of the developmental responses to warm temperature in particular. So we've published this before already, so I'll go over it quite quickly, but we found a role for the phytochromes as temperature sensors. And I'll just highlight that because it's important for thinking about plant development. And we got into this like a lot of other people did, because we saw that hypocotyl elongation in plants is a great assay for measuring the temperature response. So here you've just got 4 seedlings that are grown at 12, 17, 22, or 27 degrees. And what you can see is that as you increase temperature, you have a huge increase in the elongation of the hypocotyl. So this is really a great assay for us to look at how the plant is perceiving temperature. And a postdoc in the lab, Jae-Hoon Young, saw that when you took mutants of the phytochromes, you basically, even at 12 degrees, you have a constitutively long hypocotyl. So a phytochrome mutant essentially resembles a warm-grown plant. And so we said, okay, yes, this is a case for hypocotyl elongation, but this is the case, for the whole plant. So we said, what if we look transcriptionally? Does our phytochrome mutant transcriptionally resemble a warm-grown plant? And so this is a bit of a busy slide, but if you just focus on the middle 2 rows in this slide, this is just looking at gene expression in Landsberger vector, so our wild type control, at either 22 degrees or 27 degrees. And these bars on the side show you the day-night cycle. So these are plants that are grown in short days, and so they have a long night and a short day. Okay, so black is day, black is night, and white is daytime. And basically what we've done is we've taken samples about every 4 hours. We've performed RNA-seq, And then we've clustered for gene expression according to major patterns of gene expression. Okay, so we have different clusters here, and these are just sets of genes that are behaving the same way. You don't need to look at all these clusters, but if you just focus on cluster 16, so this cluster here, what cluster 16 is showing us is that during the nighttime, these genes are green, so they're highly expressed at 27 degrees in wild type, but they're not induced in 22 degrees. So here you have a set of genes that's specifically responding to temperature at night, and it's highly expressed at 27, but not 22. And there's a similar cluster 20, which is also expressed at 22 degrees here, at 27 degrees here, but not at 22 in wild type. So these are two key clusters, 16 and 20, and which are simply responding to temperature. So these are like outputs of this particular thermosensory pathway. And so we then said, well, if phytochromes are important, they should have a disruption in the induction of these genes. And that's very much the case. So if you look at cluster 16, this is already now constitutively expressed in the phytochrome A, B, C, D mutant. So if you take away all phytochrome activity, you have a constitutive expression of the genes that are otherwise temperature induced. And we can go in the other direction. So we can make a constitutively active phytochrome, and this is called YHB, and it's a mutation which was created by Clark Lagarius and UC Davis. And when you have constitutive phytochrome signaling, you then basically, you have constitutive repression of this whole cluster 16. So it's off at 22 degrees, which is what you expect, but it's also no longer induced by 27 degrees, the way it is in wild type. So it's purple when it should be green. So basically that's telling us that this cluster is really responding kind of very comprehensively to phytochromes. And so what are phytochromes doing in temperature sensing? So we all know about phytochromes being essential red light sensors. So phytochromes are in an inactive PR state, and when they're exposed to red light, they become activated, so they have a conformational change, and they enter what's called a PFR state. And in the PFR state, phytochromes are able to bind to and repress the PIF BHLH transcription factors, and they do things like repressing elongation growth, germination, and flowering. And far red light very rapidly inactivates the PFR state to the PR state. So there's this balance, depending on the spectrum of the plant is experiencing, between inactive and active phytochrome. And this interconversion, photoconversion, takes milliseconds. So it's a very fast response. But during nighttime, something quite interesting happens, and it's been known since the 50s, that during nighttime, if you have active PFR, it will slowly revert to the inactive PR state. But this takes hours, not milliseconds. This process takes hours. And people have been curious what the function of this so-called dark reversion stage is. And so we went and checked with our collaborators if this is actually responding to temperature. So is the dark reversion proportional to temperature? And so that's very much the case. So if you just plot the half-life of PFR against temperature, what you see is as you increase temperature, you rapidly decrease the half-life. And this is a log scale. So basically at 4 degrees, the half-life is greater than 10 hours. Whereas at 27 degrees, the half-life is less than 1 hour. And this fits very closely with all the modeling we did and all the physiological measurements we were able to do in phytochrome A, B, C, D mutants and their temperature responses. So the reason, this is the model then, the reason a plant grown at high temperature looks a bit like a phytochrome mutant is because it is essentially behaving like a phytochrome mutant, at least during the nighttime. So during the daytime, you have photoconversion, so you have fully active PFR. But during the nighttime, there's no light for photoconversion. So PFR starts at about 100%, but then will decline during the night. And at high temperatures, this decline is so rapid that by the middle of the night, you're already running out of active PFR. And so the plant's essentially resembling a phytochrome mutant. And that's why many thermomorphogenesis phenotypes resemble phytochrome mutants. Okay? And this is just a nice summary from a nice review by Quint and colleagues. And it's just sort of interesting from an evolutionary perspective. If you think the phytochromes have evolved to be these absolutely massively important sensors of light which I integrated into basically all the developmental and physiological processes in the plant. So almost everything is affected by a phytochrome mutant. And that makes sense because light is so central to plant growth and plant survival. So it looks like what temperature has done is it's co-opted this amazingly central and fundamental sensory developmental regulator and added on a temperature component. So plants can now multitask and use phytochromes both for light and temperature signaling. So it's kind of interesting from an evolutionary point of view. And the really nice thing is you can separate these two functions because, as I said, the mutation in YHB doesn't affect photoconversion at all. It just changes the dark reversion rate. So this is also quite interesting. So during the day, phytochromes can do something completely different from what they're doing at night. Okay, so what are other ways? So we knew that phytochromes are not the only temperature sensors in plants. So what are other ways that plants can sense temperature? And this is recent work we published, which is looking at a complex called the evening complex. The evening complex is a key component of the circadian clock. And it controls the expression of PIF4. So you probably heard of PIF4. It binds to phytochrome, and PIF4 is a major transcription factor that activates many thermomorphogenesis pathways in plants. Okay, and what I've said just now is that phytochromes control PIF4 post-translationally. So phytochromes bind to and inactivate PIF4 protein. But PIF4 transcription is actually also controlled by temperature. So when you increase temperature, you get more PIF4 expression. So temperature is interesting. It increases the expression of PIF4 and it inactivates the phytochromes, so you have more PIF4 protein activity. Okay, so we really wanted to know how is it that the temperature can activate the expression of a gene. And so what we found was that PIF4 is controlled transcriptionally by this complex called the evening complex. And at lower temperatures, the evening complex, which contains 3 proteins, ELF3, ELF4, and LUX, which is a transcription factor, and recruits the other two targets genome-wide. What we found is that at low temperature, this complex is completely active in Arabidopsis. So the complex is active and it represses gene expression. So you have a low expression. So targets of the evening complex include PIF4 itself. So at low temperatures, PIF4 is repressed and not expressed. When you increase temperature, you have a big pulse of PIF4 expression. And that occurs because the evening complex falls apart. And you can actually see this genome-wide. So this is just a ChIP-seq where we've done a pile-up of ELF3 binding to all its major targets genome-wide. And in green, you can see the amount of binding at 22 degrees. And when we increase the temperature to 27 degrees, within hours, there's a massive drop in the binding of ELF3 to its target. So you go from this level down to here, okay? So we said, okay, this is interesting. ELF3 is responding to temperature, and it might be direct, and very rapidly. So how is ELF3 able to do this? And when we looked in the sequence of ELF3, we found there was a very interesting polyglutamine tract. So there's a string of glutamines in a region of ELF3. And what we found was that when you change the length of these glutamines, you change thermal responsiveness. So this is just measuring hypocotyl length, and hypocotyl length is smaller when you have a shorter polyglutamine. And as you increase the length of this tract, you effectively appear to make the plants more sensitive to temperature because the hypocotyl elongation increases with the length of the polyQ. We thought that's interesting. But these plants are still responding to temperature. And so we looked more broadly at what might be going on, because even if you have no polyQs and no glutamines in the polyQ, you still respond to temperature quite well. Not as well as a long polyQ, but you still respond. And when we looked, what we found was that the glutamines are actually within the middle of a prion domain or predicted prion domain. So even Q7 has a big prion domain, predicted prion domain in this region of the protein. And actually, as you increase the length of the polyglutamines, you increase the length of the prion domain, which is what you'd expect because glutamines are very priogenic. So we thought, well, that's sort of interesting. So maybe the prion domain is basically controlling this temperature response, okay? And so we said, well, can we find ELF3 in other species that might be more adapted to high temperatures? And one good example of a plant that's adapted to high temperatures is Brachypodium. So Brachypodium has basically, grows throughout the Fertile Crescent, throughout the sort of, Eastern Mediterranean, and it's very acclimated to high temperatures. So it can grow very well in much higher temperatures than Arabidopsis can. And what we found when we looked in Brachypodium distachyon, which is relatively heat resistant, we found that it contains a copy of L3, but in this case, the prion domain is almost completely lost. And so this is just a plot like the last one showing you whether you're predicted to be a normal protein in black or whether you're predicted to have a prion in red. And here you can see the red does not go over about 50%. So at least according to this model, the prion domain in Brachypodium L3 is almost completely lost. So we put these versions of L3 back into an L3 mutant. So an L3 mutant plant you can see here is very spindly and early flowering. So it has a constitutive warm temperature phenotype. This by comparison is Columbia, which is early flowering at 27, but it's still much more robust than these L3 mutants. If you overexpress Arabidopsis L3, you don't have any effect. So overexpressing Arabidopsis L3 in the L3 mutant background basically makes the plant look like Columbia itself. So it rescues the L3 phenotype, but it's still early flowering like Columbia. But then when you put in L3 from Brachypodium, you now create this plant here, which is completely rescues the L3 phenotype. So this has a normal clock now, but it's over-rescued almost, because basically what we've done here is we've added a version of L3 which appears to be more or less insensitive to temperature. So it basically, the plant now is unresponsive to warm temperature altogether. And we can see that with just looking at flowering time. So at colder temperatures, plants are very late flowering. When you increase the temperature, plants are early flowering. This is just looking at leaf number. And this is true for overexpressing Arabidopsis L3. This is true for Columbia itself. If you take Columbia, you increase the temperature, it flowers earlier, so you have fewer leaves. L3 mutant is flowering early all the time because it's basically got constitutive activation of the high temperature pathway. But when you overexpress the Arabidopsis, the Brachypodium L3 in Arabidopsis, you basically now have plants that are very late flowering. So here, this is 27 degrees, and they're almost as late as plants growing at 22 degrees. And it seems to be specific for the PRD. So if you take the Arabidopsis L3 promoter and you use that to express Brachypodium L3, you're late flowering. And then you can be even more extreme. So you can take the entire genomic cassette and just replace the prion domain. So this is an Arabidopsis promoter driving the genomic region of Arabidopsis L3, simply replacing the prion domain region of Arabidopsis with that of Brachypodium. And now you're also very late flowering. So this domain seems to be necessary for controlling the protein and its temperature response. And so at this point, we've sort of got interested in reading up work on phase change. So this is an area, I don't know if you're familiar with it, but Tony Hyman in Dresden has really pioneered and pushed this area of biology. And there's a lot, I think, of interesting papers coming out in this area at the moment. And what Tony Hyman is essentially saying is that you should think about proteins as existing in at least two phases sometimes. So we're familiar with what's here on this is a single phase. So this is a protein that's basically soluble in the cytoplasm, and it's soluble in essentially an aqueous phase. But what can happen with certain proteins is that they form liquid droplets. So they enter a sort of basically a solution of themselves. So instead of being in a solution of water, they're in a proteinaceous solution in these liquid droplets, which are very high concentration. And they can form under particular biophysical conditions, and particular proteins have a tendency to be able to form these liquid droplets. And we wondered if L3 was doing this. And so we looked in plants, basically we looked in the root with GFP fusion of L3. And what we can see here is that when you have an L3 in Arabidopsis expressed in the root under the native promoter, 17 degrees, it seems to be nuclear, but it's fairly diffuse throughout the nucleus. And when you increase the temperature, what happens is you form these little speckles within the nucleus. And you can see these very clearly here. And when you express a Brachypodium L3 fused to GFP, you lose this behavior. So here, even at 35 degrees, you see no speckle formation of Brachypodium L3, whereas you do with the Arabidopsis L3. And this seems to be a very sort of basically a universal property of these proteins, because if you could take these proteins and you can express them in yeast, and you can also drive formation of these liquid droplets. And yeast doesn't have the evening complex. It doesn't have any of the other components. So for yeast, L3 is a completely new protein, and it doesn't have any other components of the machinery, but it still makes liquid droplets. And so we can also see this in vitro. So if you take this part of L3 and you express it in E. coli to purify the protein, you can also see that this region of L3 is sufficient to make liquid droplets in vitro. So you can express a protein in vitro, you can purify it, and then you can change the temperature. And what happens is, as you increase the temperature, it goes from being soluble to making these liquid droplets. And this you can simply measure by measuring absorbance of 440. So we're just measuring cloudiness of the solution. And as you hit a certain threshold temperature, around 27 degrees, you can see nearly all of the L3 goes into these liquid droplets. So we think this region of L3 is simply behaving as a temperature-dependent sensor. So at lower temperatures, L3 is active because it's in the completely soluble phase and is able to form this evening complex, which represses gene expression. And at high temperature, L3 is sequestered into liquid droplets and is unable to repress gene expression. And the nice thing about this is it's fully reversible. So if you take these droplets at 27 degrees and you lower the temperature, they become soluble again. So it's a very nice way to very rapidly switch your protein on and off in response to temperature in a reversible manner. Okay. So that's it for me with the evening complex, really. But just to summarize, the evening complex is very interesting from the point of view of adaptation because it basically is a major part of the circadian clock, and it represses many key genes involved in photosynthesis, circadian clock, adaptation to cold stress, and growth itself. So it's a sort of very interesting example of how, again, a component in the plant of the circadian clock has acquired a temperature responsive behavior by, in this case, it looks like acquiring the prion domain. But not all plants do this. So in Brachypodium, it has almost no prion, and consequently, Brachypodium responds much less to warm temperature when we look at flowering time, for example. Okay, and just to zoom out a bit, it's sort of remarkable to us in a way, but if you look at thermomorphogenesis, basically we now have, so there's another pathway involving an RNA structure, which I won't talk about today because we don't have time, but we found an RNA structure that responds to temperature and controls a gene called PIF7, which is also binds directly, it forms a heterodimer with PIF4 and also controls thermomorphogenesis. So in the thermomorphogenesis pathway, there are at least 3 very discrete thermosensory events that contribute to all the responses we see, developmental responses we see at high temperature in Arabidopsis. So it's quite interesting that this one pathway has acquired 3 quite distinct and discrete temperature sensing events. Okay, so that's like the warm temperature response. So I don't know about you, but we recently had a period of extreme cold weather. So this was just where I live a few weeks ago. We had basically masses of snow for like 4 days. It was fantastic. And it got extremely cold. So it went down, so here it went down to -26. And just the other day, it went up to +25. So basically within Brandenburg, we've had like a temperature gradient now of 50 degrees within a few days. So it's kind of amazing how plants cope with this. And so, of course, one of the really important ways plants cope is by having a cold adaptation pathway. So Arabidopsis and most other plants have evolved an ability to protect themselves against freezing stress. And this is something we've also been interested in for a while. So really classical experiments by people like Mike Tomashaw show beautifully that you have cold activates the expression of genes such as the CBFs, and the CBFs are sufficient to confer cold tolerance. And so this is work by a PhD student called David Schofer in the lab. And David wanted to know how plants sense cold temperature. And this is actually on bioRxiv. So if you want to read the paper in detail, you can. But I'll just summarize some of the findings because I think it's quite neat. And so what David saw was that protein translation in the cell is very temperature dependent. So if you measure the rate of translation on the ribosome of our proteins, what you see is the rate of translation increases very linearly with temperature. At low temperatures, you have almost no translation. And then as you increase temperature, you get much more translation. You can do this looking at the production of luciferase in this case, which is what David did. And you can also look at the incorporation of things like puromycin. So you can look at the incorporation of analogs into proteins to measure protein synthesis genome-wide. So what you see, however you measure protein production in the cell, you see protein production goes up as a function of temperature, right? And so what David wanted to know basically was, is translation rate giving input into the temperature response pathway? And the nice thing with translation is you can modify it with drugs, right? So you have drugs like cyclohexamide that inhibit translation very strongly. So you can very specifically knock out translation and very quickly in the cell, even at room temperature, okay? And so what David did was he said, does inhibiting translation interfere with the cold temperature response of the plant? And this is just a heat map showing you the induction of the first wave of gene expression in response to cold. So these are plants that are shifted to cold for a quarter of an hour, half an hour, 1 hour, 2 hours, 4 hours, 6 hours, okay? And this is in wild type. And you can see this cluster, the red cluster here, cluster 1, are the first genes that are very strongly upregulated by cold in the plant. And this is what we could call the first wave of cold-induced gene expression in Arabidopsis. And it contains all the classical candidates, such as the CBFs, okay? And what David saw was that all these genes that are specifically upregulated in response to cold can be specifically upregulated simply by adding cyclohexamide and inhibiting translation, okay? And so these are plants that are kept at 22 degrees. So there's no cold shift here, but we've got massive upregulation of all the cold-induced genes. And he showed this with qPCR, and he's verified this as well. And it's a very clear pattern. So CBF1, CBF2, and so forth. So far, they're all basically strongly induced by cyclohexamide. There's a second wave of gene expression in the cold. So basically, these genes here in the later clusters are genes that are induced by the CBFs. And those genes are not induced by cyclohexamide. And that makes complete sense, because for these genes to be induced, the CBFs have to be transcribed and translated. And of course, when you add cyclohexamide, you no longer have translation, so you cannot induce the second wave of cold-induced gene expression. So again, this suggests that it's a very specific effect of translation on the first wave of gene expression. And key factors in the cold response are the CAMTA transcription factors, and those are basically activated by cyclohexamide. So this is a ChIP-seq experiment where we're looking at the binding of CAMTAs to promoters. So these are the CBF. This is CBF operon here. You can see you've got the 3 CBFs, which are in an array on the genome. You can see cold induces a strong binding on CBFs across the whole array here, compared to mock treatments. But the cyclohexamide treatment in the warm is sufficient to basically trigger activation of CAMTA binding on all the CBFs, suggesting that something to do with translation is directly activating the CAMTAs, which are activating the first wave of cold expression. And there's, as you'd expect, a huge overlap between those genes that are induced by cold and those genes that are activated by cyclohexamide. And this is also true in terms of degree. So the relative strength of translational inhibition correlates with the degree of activation of the cold response. So David has basically assayed dozens of drugs here, all of which are known to inhibit translation to different degrees. And you can see the extent of inhibition of translation as measured by puromycin uptake basically anti-correlates very well with the degree of CBF2 expression. And so to see if this was really a direct effect of cyclohexamide on translation, David created mutant plants where he expresses a mutant form of a small ribosomal subunit where cyclohexamide normally binds. So in yeast, people did a mutant screens for yeast that are able to survive cyclohexamide treatment. So if you have mutations on the subunit that's bound by cyclohexamide, you can create mutants that don't bind cyclohexamide and are no longer inhibited by cyclohexamide. And by putting the same mutation into a corresponding small subunit of the ribosome in plants, these mutants called CYHR1 no longer bind cyclohexamide. And so they're able to grow in cyclohexamide, whereas mutants with wild type, of course, are killed by cyclohexamide concentrations above 3 micromolar. And these mutants, though, are completely happy growing in cyclohexamide because the ribosomes no longer bind cyclohexamide. And in this case, we lose the induction of CBF2 in these mutants. So wild type has a strong induction of CBF2 in response to cyclohexamide. But when you treat these mutants with cyclohexamide, they no longer induce CBF2. Okay, so that we can cut the cold response in these plants just by changing the ribosome. And finally, we wanted to know how this is happening. And we think this is happening through calcium, because it's well known that when you treat plants with cold, you have an increase in intracellular calcium. And David can he can recreate this kind of exactly simply by treating with cyclohexamide. So this is in black you see mock control, and then in red you see cyclohexamide trace. So this is just measuring intracellular calcium using Corin in this case. And what you can see is there's a big increase in intracellular calcium in response to cyclohexamide. And this is completely abrogated when you mutate the small ribosomal subunit that no longer binds to cyclohexamide. So this is our final model. We think what's happening is that cold is inhibiting translation. Slowing down translation results in increased intracellular calcium, and that's sufficient to activate the Kempler transcription factors, which bind directly to the CBFs and induce cold expression. Okay, so I'd just like to thank everyone who's done all the work. So I talked about the work of Jae-Hoon Young, who's now a professor in Korea, who did the work on the cold signaling, on the phytochromes and the evening complex, along with Daphne Ise, who did an amazing amount of bioinformatics and helped us with the analysis. Martin Balcerowicz did work on PIF7, which I hadn't had time to talk about, and David Schofer did the work on cold signaling. So we're now, as I say, we've now moved. So we're now at the University of Potsdam at the Leibniz ITZ, and this is the new group of people. So thank you very much for listening, and I'm happy to take questions.
Thank you very much, Philip. It was really a great talk and great research. So I would like to open the floor for the questions. Do we have any questions? If you have questions, you can also activate the camera. Yes, Heribert, you can start.
Yeah, thanks, Phil, for this nice talk. I have a question in terms of the ribosome. So you say that it's basically a sensor to cold stress. How about heat stress?
Right. Yes. So that's sort of very interesting question. So there are, so it's certainly possible. So there are connections between, certainly in mammalian cells, there was a very nice science paper showing that many targets of the heat shock factor transcription factor are affected in cyclohexamide treated cells. And the way David actually got cyclohexamide in this screen in the first place was by looking for things that change the expression of HSP70 when you treat seedlings that have an HSP70 luciferase reporter. And what we see, the reason we got interested in this process in the first place was that when you treat cells with cyclohexamide, you do have a shutdown of the heat stress response. And that's how we got into this. And then we saw the cold response is activated in this background. So you're 100% right. There is a connection between heat and the ribosome. I think it's more complicated. So exactly what's going on with heat and the ribosome is more complicated, but I think there are ways the cell is using that information in sort of multiple ways. So one of the things you know is that when you go into very high temperature stress, you basically have a shutdown of translation. That's one of the adaptive responses. But of course, if you want to adapt to high temperature, you also need to translate the essential proteins you need for heat stress protection, like the HSPs.
Yeah. Because what you showed in your curve, that goes up to a certain point, and then I think it's around 30 degrees. You really have a very steep drop, if I remember that right? So that looks also as if there's a real mechanism of regulation, right?
Yeah, you're absolutely right. So we haven't followed it up yet, and we don't have insight enough. We don't have enough. Yeah, we haven't done anything useful, but you're 100% correct. There's a very clear response, and it sort of makes sense. So, and I think ribosomes are very interesting because if you think about adaptation, ribosomes have evolved in the organism to be active and have the right level of activity for whatever climate the organism happens to have adapted to, because translation is such a central component of cellular life. So if you want to have something which is like a fundamental sensor, a ribosome is super interesting because it has to adapt to whatever conditions that plant or animal even will be happy at.
Yeah, great, thank you.
Okay, now Stefan.
Hello, thanks very much for this interesting presentation. Actually, I've been working sort of by chance on a protein from bacteria who was sensing heat, so I was wondering how plants would do the same. So you mentioned in the first part of your presentation that we have this evening complex that falls apart when the heat rises too much. And you showed that this was dependent on the presence of a polyglutamine truck in L3, which you called the prion domain, I think, which is probably a little bit a misnomer because then you showed that it's undergoing LLPS. So I was wondering, because you showed that this L3 region was undergoing LLPS on its own without the presence of the other proteins. So how do you think this is now related to the evening complex, which contains different components to fall apart? How is the LLPS of this region linked to the complex stability?
Yeah, so great question. So first of all, I just want to totally agree with what you're saying. So we call these prion-related domains because they're predicted to be prions using basically a model that was trained on yeast data. So it's a hidden Markov model, and they've just taken proteins from yeast that form prions in yeast, and they said, okay, if other proteins contain this sequence, they're likely to be priogenic. So historically, I mean, a prion is defined as like a irreversible aggregation of proteins in a stable sort of, amyloid almost state. And so proteins in yeast that are defined as prions are like the criteria are that you can boil it in SDS and it remains basically insoluble. And as you say, what we are seeing is something which is much more gentle, which is something that can do reversible liquid phase transition. So you're absolutely right. So these prion domains, it's still very useful for us because we use the program to find domains of protein that are likely to undergo phase transition. But it also asks the question of why life has retained prions. And one reason might be that prions, as we know them from the early days, is just it's an artifact from having proteins that have become insoluble. But the most useful form of a prion for a cell is probably a protein that can undergo reversible phase change. Okay, so you've got that. So then you say, okay, well, so what's happening when we're taking part of L3 and we're doing our in vitro work on that, but actually it's a whole L3 in the context of its binding partners. That's really important. And the long story short is that we cannot get soluble full-length L3 in vitro. So that's a bit of a pain. That's how it is. So what we need to do is exactly what you're saying. We need to basically express the whole evening complex in vitro with the altered prion domain. And then we'd like to also go back and then say, okay, can we show the evening complex itself is changing its behavior in response to temperature in vitro? And so we have our collaborators in Grenoble have very promising sort of preliminary evidence that if you do band shifts with a reconstituted evening complex, the band shift is temperature dependent. So at high temperatures, the band shift falls apart. But we need to go back and do that with a stabilized prion protein to show exactly that it's that domain that's causing this effect. I'm pretty sure it is because of all the work in Planta, but you're 100% correct. I agree with you. That would be the really key sort of follow-up.
Thank you.
Okay, I think now Ikram is your turn.
Hi, Philip. Thanks for the beautiful talk.
Thank you.
It's really nice, especially the ELF3 story and the shift that you see in localization in Planta and vivo from nuclear into these nuclear speckles. So do you know what are these speckles? Are they, do you know the nature of their, because some of them are usually related to RNA splicing, to transcription, etc., etc. Do you know which foci are these ones?
That's a great question. So,
and then the second one. So
yeah.
when ELF3 is in these speckles, is it active? What happened to the complex that its binding partner?
Yes. So in this case, we think the speckles are inactive. So we know the speckles are forming at high temperature, which is when we see L3 leaves DNA. And so we think the speckles are a way to sequester L3 away from its binding sites genome-wide, which we can measure by ChIP-seq. Now, do the speckles contain L3 and Lux? That's a good question. We don't know. They may contain the other components of the evening complex in the speckle. And what is happening in these speckles? So, as you say, speckles are often associated with transcription factor factories or splicing events. In this case, because it's inactive, it might be just a way to keep L3 away from its targets genome-wide. But the other things that might be in those speckles, we would love to know. So this is exactly where, I wrote a grant recently, and one thing I'd love to do, perhaps in collaboration, would be to say, can we use, say, for example, fluorescence activated sorting to purify the speckles and do proteomics and also RNA-seq, because a lot of these droplets that form can basically contain RNA. So RNA is a great way to nucleate liquid droplets, because it can bind proteins and increase local concentration of proteins. So we have no idea, but we would love to know.
Okay, and did you try to colocalize them with all the available nuclear bodies from Petershaw?
We haven't done that. Yes, a great idea.
They're even in TER. You can just order the clones and then do your colocalization.
Okay, that's a great idea. We should do that. Thank you. Yes. Yeah, no, absolutely. I think that would be really, really interesting. And so we are seeing speckle formation for a number now of other proteins we're interested in. And what's interesting is, in relation to what your question in a way, we're also seeing proteins can undergo phase change and form speckles, and they seem to be the active form. So that's good. So we'd also like to follow some of those things up, because it's even more interesting if the protein in the speckle is in the active state. There are things called photobodies, which people have been studying for a long time, which contain Phi-B. And so we know Phi-B forms speckles, and it's a bit tantalizing because we know L3 and Phi-B interact. So it could well be that there's a connection between photobodies and these L3 bodies, but we don't have any evidence.
Thanks. Thanks a lot.
Thank you.
Okay, I think Alagozi can have a question. Hi, Ziagis.
Yeah, that's me. This is because of the computer software. Can you hear me?
Yes, thank you.
Thanks for your nice presentation and your nice work that you've been doing. I actually read one of your papers that you published last year. It's about the P7 RNA thermoswitch.
Mm-hmm.
Actually, I'm just asking for out of curiosity, do you have any other candidate genes that you're working on to uncover such a similar mechanism? And also the second question is how frequent they found in the 5' UTR of these genes?
Right. So what we haven't. Well, so there was a postdoc who was meant to come to work on HSFA2. So the other gene we found, which we were very interested in that paper, was HSFA2. and that also has a thermoresponsive hairpin. And we can show that that's having some role. And we wanted to really work out, but the postdoc, unfortunately, has been basically completely, her plans were scuppered by coronavirus. So that whole project is kind of on hold. HSRA2 would be the other one we were interested in.
Okay.
But it's hard. So the difficulty of that project is that it's pretty much impossible bioinformatically to identify these things from first principles. So if you take like all the 5' UTRs of all the genes in a genome, and you say, okay, which of these are forming thermoresponsive hairpins? It's impossible, because the prediction programs can predict almost every imaginable shape, depending on what time of day it is, or exactly what concentration of sodium chloride you're specifying, or what temperature it is. So RNA secondary prediction, you can do it, people do it, but it's very, very hard to know whether it's real or not. And so we're quite limited in our ability to be able to do genome-wide comparisons, which is what we would like to do. So the only way I think that makes sense is to actually go into other species and do RiboSeq. But we haven't done that. We're not really experts on RiboSeq. I think the way forward is to do RiboSeq. So it is quite interesting if you think about it just from the point of view of designing experiments. So a lot of people have been looking for a very long time for studying RNA secondary structure from first principles, or then even trying to measure RNA structures globally in the plant in different temperatures, and then go from an RNA structure to a physiological response. And this is just incredibly hard to do, simply because you have so many different species and so many different shapes.
Yeah, I agree with you. We tried to pull all the riboswitch candidates. I was actually working on a riboswitch candidate gene in the RNA, and then we pull all these microorganisms, riboswitches, and then basically had two approaches. One shape-based, the second one is the sequence homology, and we tried to merge all these data together and then identify a couple of them, and then I tested one, and then I'm still not 100% sure that the one that I found is a kind of like a riboswitch, but I have the data. But
Brilliant.
I was just trying to understand maybe if you have a different approach to identify these ones.
No. It sounds like you are way more advanced than we are. So what you're doing sounds absolutely brilliant.
Okay.
Thanks. I know in microorganisms, RNA riboswitches are much more well defined.
Yeah.
So I think it's actually a better system. I think in eukaryotes it's just extremely hard, but I think what you're saying is a very good approach, and I think the ideal way is you combine. I think you'd combine two filters. So I think taking a sequencing filter and a shape filter and adding them together, and then saying, okay, we're going to eliminate as much as possible, and hopefully something will be left over at the end. I think that's a great approach. I think I'll basically try and get in touch with you if we try and get this project going again. I mean, it's tantalizing. There is something there, but doing it genome-wide is hard.
Well, I'm not doing it here, but I did it in my PhD. So
okay.
Let's see. All right. Thanks for the feedback.
Yeah. Thank you.
Okay, I see we have also a question in the chat from Hanin. So the question is for you, Philip, that if you know whether both C3 and C4 plants, they have the same thermosensor mechanism.
That's a good question. Yes. The answer is I really don't know. So we're very limited at the moment in what we do. So most of our work has been in Arabidopsis until now, and we're now moving a little bit into rice and a little bit into Brachypodium. I'm not sure if the Brachypodium is a C3 plant. I'm not sure if we have any results in C4s at all. I don't think so. No. But would I expect a difference? So only in that. So one thing we're doing at the moment is we're doing quite a lot of genome-wide scanning of extreme heat adapted plants and extreme cold adapted plants and plants in the middle. We're not separating by C3 or C4. We're just saying for plants that are acclimating to very high temperatures, can we find signatures in proteins where we see PIDs that we think are controlling temperature responses that would suggest adaptation? And we think we can. So we're quite excited about that. So we think we're going to find patterns where there's temperature dependent differences in proteins depending on where you grow. In very analogous to what we see with Brachypodium, but on a sort of bigger scale. So we are very interested in that. And so we're sort of following that up. And that's just it. For us, it's sort of interesting because I think a lot of people have been doing this genome-wide for a very long time, trying to find signatures of evolution or adaptation for heat or cold stress genome-wide using a very unbiased approach. And I think that's actually been relatively hard. But I think this is a case where once you know a little bit of the mechanism, that means you can then go in and ask a slightly more directed question in a broad background and then say, okay, do we see correlations? And so far we do. But I can't, so T4, we'll have to see. It's a great question.
Okay, so Mike, and after Mike, I will ask my questions.
Can I ask?
Yes, go.
Sorry. So I had some questions that they were already questions and replied from before, but I would like to ask about this difference in temperature that you have between the extreme higher and the extreme lower. You said that it's about 30 degrees or 40 degrees between winter and summer, day and night, in the extremes at least. So if but I can say that here in the desert, for example, we have much higher differences in the summer and the winter, in the day and the night, but maybe it's 50 degrees, maybe even more. So I think that plants there and plants here, of course, different plants, they have different adaptation. They have adopted different adaptation mechanisms. So if this is a universal way to control also some developmental or responses or developmental or abiotic response to abiotic stresses, for example, if this huge amount of difference in the temperature between that the plant is continuously undergone, if it is important for the output that they can have either developmental or in the abiotic responses.
Sure. So yeah, desert plants are absolutely amazing, right? So their ability to tolerate these swings in temperature is super interesting.
Because here we're not talking. Here we're not talking about cold. We are talking about less heat. So 10 degrees here is very much cold in England, right. And it's not.
Exactly. So that was a bit like what I was saying. So for one plant, 10 degrees is a heat stress, and other plants it'll be a cold stress. So in the desert, 10 plants will start to be a cold stress. So well, I mean, it's kind of hand wavy, but it seems to be clear that you have basically a certain range which your cellular processes can be active over. And it looks like most organisms, most plants, they basically make a choice. They're basically, so a desert plant will be able to tolerate super high temperatures, and then you can get cooler at night. But it would probably die if you took it down to 10 degrees for much. So we're using rice, for example, to do quite a lot of this. And so what we're seeing with rice is that at 10 degrees, basically 10 degrees for rice is cold stress. And so, and what we see is we see activation of the same things we see activated in Arabidopsis. We see them activated by rice at 10 degrees. And so plants, they're able clearly to set a certain range of which they're happy at. And when you diverge from that range, they then activate stress responses. But where you activate the stress response clearly is affected by where you're adapted to grow in.
So we would be able to talk about an adopted thermal switch between higher and lower temperatures. So I would like to ask also a second question, if I have time, about before. If the mutants have some developmental outputs by means of thermosensitivity, understanding temperature changes, and if there are developmental outputs, and if there are developmental outputs that they are controlled by P4, how much specific are these ones? And by means not only from temperature, sensing, but also from salinity temperature, from salinity sensing or humidity sensing or any kind of other abiotic factor is involved in this.
Sure. So a key output of PIF4, one of the things is flowering. So what we noticed a while ago was that a PIF4 mutant has a much reduced thermal induction of flowering. So at 22 degrees, nobody notices the phenotype because PIF4 is not very important at 22. But when you go to 27 degrees, PIF4 mutants are later flowering than wild type. So PIF4 is feeding into flowering for sure, which is an important developmental response. And that basically, the PIF4, it really interacts with a light quality, obviously phytochrome signaling, and so on and so forth, and hormone signaling. The connection between salinity or drought is really hard. We haven't done anything there. There is some very nice work showing in Adaptation that drought can influence flowering time, but I don't think that's PIF4 mediated. So the intersection of heat stress with things like salinity and drought, massively important, super physiologically relevant. We have not really touched it, but I think that's the future. I think there's some very interesting work to be done there.
Okay. Thank you very much.
Thank you.
Okay, now I can ask my question. So I would like to ask you, Philip, about this cold, let's say, response or cold sensing that is quite intriguing that cyclohexamide can kind of copy the response, at least at the transcriptional level, from the cold responsive genes. So I was wondering, because cyclohexamide inhibits the translation, have you thought to check if they are stress granule form under cold stress conditions?
Right. Yeah, that's a good question. Gosh, yes. Yes. No, so that could well be the case. You could well imagine that cold stress will cause a change, particularly with things like phase change, which are very temperature dependent, could well be causing something like that to occur. And so we haven't looked, but I think that's an awesomely good question. We should try that. Because fundamentally, we are mystified by how it is that a slowdown of translation, either with a chemical or with cold, how this slowdown triggers the calcium response. And that is something we're really curious about. We don't have a good understanding of that at all yet. But that's something we're very keen.
I think there might be dysregulation of the phase change. That might be the answer to the question.
Okay, I have to talk to you another. Yeah, so that sounds awesome. Yeah, I like that idea.
And second question, I mean, Ikram already asked, but about the speckles in the nuclei, in the nucleus, you would like to extract them and identify the components?
Oh, I would love to, yes. Or collaborate with someone who can. That would be even better, probably. We don't have a strong proteomics background, and we're a small lab. So I'd really like to collaborate, ideally, with someone who's interested in that, because I think not just the L3 speckles, but we're now seeing. So one of the questions we've had is whether this is actually. So with the phytochromes, you can say, okay, phytochromes is interesting. They're doing dark reversion, but this is a very specific thing because phytochromes are the only molecule in the plant that does dark reversion. There's no dark reversion doesn't exist. So the big thing for us has been, can we find a mechanism that's more universal than dark reversion? And so with the PRDs, we're hoping that this is the case. So it's not just L3, it's other things that are also doing this. And as I said, we also have speckles where we have activated protein. And so we would really love to do mass spec analysis of those and say, okay, we're having phase change, and phase change is enabling all these components to be assembled together, essentially, right? So what is if you, without being too philosophical, right? What's the purpose of phase change, right? So we're obviously it seems to be controlling the event, but if you have a factor and that's undergoing phase change, why is this event causing either activation or inactivation? And you are 100% on the money that until you purify and do the proteomics, it's meaningless. So that is 100% exactly the number one experiment to do.
Yes, I completely agree with you. Well,
Yeah.
but then we should talk soon. I will contact you again.
Cool. Yeah, I'd love that. Yeah.
Okay, I think we have still time for the last question, and I see there is one more in the chat from Rashid. So the role of the evening complex in the heat stress is interesting. I was wondering if you are also investigating its role in other abiotic stresses. So did you check, for example, salt stress in the context of evening complex?
That's a very interesting question. Yeah, no, we have not done at all. Yep, but it's a very interesting question. Yep, we have not done. So salt stress is interesting because it will, yes, it could potentially change the osmotic pressure. And there was a very nice recent paper, so this is not my work at all, but there was a very nice recent paper from the lab of Jose Dineni in Stanford. And they created basically a sensor that measures, I think, turgor pressure, just by measuring so basically it responds to turgor pressure, and you have this inducible phase change. And so you could well imagine that something like salt stress would influence concentration, and concentration will influence aggregation and phase change. So there could be something going on there. Yeah.
Okay. Do we have any more questions, or I think? Okay, I think we had already a lot of questions, and I think it was a nice discussion. So thank you again, Philip, for joining us and sharing with us all the research. Oh, Rod is appearing.
He might be clapping. Oh, Rod is coming for the...
No, I'm just getting on. We're going to have a kind of a discussion afterwards. I just wanted to introduce myself. Hi.
Thank you, Rod. Hi.
Great
Okay,
talk.
so
Awesome.
then thank you again also to everyone for joining, and wish you a good weekend.